Mitchell Warren. I direct AVAC and delighted to welcome you here. And we do have a really exciting conversation today. So we don't want to wait too long, and we are recording this. And for those who join late, they'll only miss a few minutes of me droning on before I turn it over to our two main attractions. So it's probably just as well. Um, it is a remarkable moment to state the obvious, I think, to everybody on the line. No matter where you live, no matter who your elected officials are, and no matter what country you're in, we all know that we are going through one of the most dramatic and, for some people, traumatic moments in history, really, or at least in my lifetime. Um, and yet, we also stand at this moment in HIV where we have both huge challenges, huge disruptions over the last 5 months, particularly, but also huge opportunities. And the webinar today is really to unpack both of those things, in fact. We're really looking at what is one of the most exciting opportunities in HIV prevention, I would argue, in 44 years, injectable lenacapavir for PrEP, as a case study for what is needed to create innovation, to create opportunity to prevent infections and to deal with pandemics, not just for HIV, but really for global health. And really believe that what you'll hear today, everything from basic science through to clinical research and then looking into the future, really does uh, give us marching orders, both to make good on the promise of lenacapavir, but also to make good on the promise of basic science and global collaborations. I'm going to do a very quick introduction of just stage setting around lenacapavir. I'm then going to turn it over to two fantastic presenters, and then we're going to open it up for conversation. I want to encourage people to send questions. We got a lot of them in advance. I'm already going to try to filter what we got. We will look at the chat to the degree that time allows. We will also open up for people who want to raise their hand. And so I'm just going to jump right in. We are recording this call, and by staying on, you're consenting to that recording. It will be posted on the AVAC website and on PrepWatch, so that people who aren't able to join us at this moment are able to hear the conversation. So I'm going to start, really just to set the stage about and why we wanted to have the conversation about the scientific journey of lenacapavir. This shows what's happened over the course of now, if you can believe it, 35 years. And you can see beginning in about 2004, the declines in new infections since 1995. But we are still far, far, far off the 2025 target. And of course, we are sitting here in the middle of that year. Even with the advances 13 years ago, the approval of oral PrEP, and then subsequently dapivirine vaginal ring, injectable cabotegravir, and very soon injectable lenacapavir, we were off course on January 19th. And it is safe to say that in January 20th and since that point, we are further off track. So we've got a lot of work to do to both sustain what had been done, but also to accelerate to that target. This is from the HIV Modeling Consortium, just to give you a sense of what happens. This is just looking in East and Southern Africa. What happens if we don't invest in PrEP, voluntary medical male circumcision, and condoms? The blue line would happen with current levels. Again, still far too many infections, but the orange line showing what happens if we don't change. Of course, this is all happening at the most remarkable moment in prevention. The price of cabotegravir is beginning to come down slightly in 2026. Of course, lenacapavir soon, within days of approval. The launch of MK8527 oral tablet for PrEP, about to start later this year, and the dual prevention pill, the first dual purpose product since the female condom in 1993, might soon also be pre-qualified by WHO. And this is when we should be accelerating to our goal, not trying to save vestiges of what happened. You all know this or you wouldn't be on the call. The time is now, and I'm excited that one of our speakers, of course, is Linda-Gail Bekker, who, to a rousing standing ovation about 11 months ago, provided the results of Purpose 1. We then, a few months later, Purpose 2, and then a lot of challenges of what do we do to make good on the science. And again, you'll hear about the science in just a minute. Quick time check. There was a lot of excitement from July till September with the results announced. December, Global Fund and PEPFAR made an announcement that was an ambition to get at least 2 million people onto Len within the first 3 years. We were pushing for even greater ambition, but even that 2 million was far higher than what we've seen with any PrEP introduction in the last 15 years. And then we saw a rapid succession of activity. The FDA submission, World Health initiated the guideline process. The European Medicines Agency is now also reviewing an application. And we stand here today on June 11th, and the date that is expected to have a result no later than June 19th. It could happen earlier, but no later than June 19th. We believe FDA approval and WHO planning at IAS in Kigali in July guidelines. Huge question mark, of course, because of all that's happened with funding freezes, legal action, widespread programmatic disruption of what happens with Len. And we're not going to spend a lot of time on that topic today. Obviously, issues of access have always been critical, and they're going to be even more critical once this is an approved product. But we really wanted to focus on what we need to be doing. I will say just a few hours ago, hopefully in your inboxes, you will have seen a brand new report from AVAC looking at an analysis of what's happened in PrEP for 7 years, or in 7 countries over about 10 years, to really look at laying the groundwork for how to get it right this time. And we still hope that we can get it right, even in the context of a very different political environment. And a number of lenacapavir documents here, just so you see them at avac.org/lenacapavir. But all that happens in this context, and I wanted to highlight 3 trackers that are now online. I'll start at the bottom, one looking at STIs and how funding of STI surveillance and research is at great risk. There's an analysis of the PEPFAR stop work orders on PrEP programming, particularly, that make lenacapavir all the more challenging. But for purposes today, I think the most relevant of these articles is the HIV prevention R&D at risk. Because even since January 20th, as USAID was being dismantled, as PEPFAR was being disrupted and changed, the research enterprise was simultaneously being really exploded. And we see this each and every day with cancellations of grants, everything from basic science to clinical research to clinical infrastructure, to working with essential populations, not just in HIV, but in global health generally. And we wanted to do this conversation to understand that although we're all looking at the access to injectable Len going forward, that if we don't ensure that HIV prevention R&D gets back on track, we run the risk of not having the next lenacapavir, the next cancer treatment, the next Alzheimer's drug, because basic science and international collaborations matter. And they've made lenacapavir possible. And we really wanted to have this conversation to be able to focus on that. So we wanted to set the stage with that. And it's my great pleasure to start this off with a presenter who is becoming more famous, perhaps, than he thought he might in the last few months, and has won and is winning awards, it seems like, each day. And it's Wes Sundquist. And we think a lot about the PURPOSE trials. We hear a lot about Gilead and their development of this product. But of course, every innovation, as we say, every success has many parents. Failure is an orphan. And certainly Wes is not the lone father of understanding capsid, but really was one of the pioneering researchers, supported very importantly by the National Institutes of Health for decades, to really unpack and understand the capsid. Wes is the distinguished professor and chair of biochemistry at the University of Utah. And his research stems from his lab's work on HIV assembly and replication, and includes the biochemistry and cell biology of the endosomal sorting complexes required for transport pathway. Now, I think that might be the first time I've ever described that pathway. It has an acronym of its own. That is new language for AVAC, but without it, we would not be sitting here on the cusp of FDA approval of lenacapavir for PrEP. I do want to just highlight 2 other quick things. One, just a few weeks ago, Wes was named, along with a collaborator at Gilead Sciences, on the 2025 Time 100 list of most influential people. Not many biochemists make it there. And then just last week, along with dear friends Yvette Raphael from Advocates for Preventing HIV and AIDS in Africa, and Mopli Das from Gilead, the 3 of them were awarded the prestigious Mani Blahnik 2025 Breakthrough of the Year Award from the AAAS, the publishers of Science magazine, who named lenacapavir the breakthrough of the year. So there's a lot of excitement, and Wes, we're super excited to have you here to give us the history behind why we should be excited for an approved product.
Thank you, Mitchell, for that great introduction. And hello, everyone. Can I start by making sure you can hear and see me?
You are loud and clear, and a beautiful slide to start.
That's great. Thank you. So as Mitchell said, I'm going to tell you about the development of lenacapavir, and I'm a basic scientist. But before I tell you about the development of lenacapavir, I have to tell you something about its target, which is not one of the most famous targets in HIV, and that's the HIV capsid. And so just as a reminder, viruses are parasites on cells. And so they're much smaller than cells, but they're still quite sophisticated, and they're able to leave an infected cell and infect a new cell. That's how an infection spreads. And my great colleague here at the University of Utah, Janet Awasa, has developed this molecular animation that shows our current understanding of the process of viral exit from an infected cell for HIV. And so below the line is the infected cell. This is the limiting membrane of the cell, and this is the outside of the cell. And what you're going to see is that the viral structural proteins that are shown here in yellow and orange have the ability to assemble into a spherical particle that can bud from the cell and also pick up the spike proteins. These are famous as the envelope proteins that are the targets, for example, of vaccines. And they're going to make a particle that can then go on to infect a new cell. So HIV is what we call an RNA virus. That means its genome is made of RNA. That's shown here in blue. You can see the structural proteins called GAG that are assembling underneath the membrane. Viruses are interesting to study because they use host pathways, including human proteins, to do some of their things. And so that's illustrated here by these turquoise proteins, which are the escort proteins that Mitchell mentioned. And together they make a particle that leaves an infected cell. So these particles are being produced, but they're not yet infectious. And they're not infectious yet because this pink protein, which is called the protease, which is the target for protease inhibitors, needs to be activated, and it needs to cut the structural proteins into smaller pieces. And one of those pieces is called CA, and that's shown here in yellow. And it's the CA protein that assembles into the unusual conical capsid of the virus. So when our lab started our work, it was in the early 90s. It was clear that HIV was a major health problem. An exploding number of men were HIV positive and were dying in the US. But we're also driven by curiosity because the CA protein was able to assemble into this conical capsid. And cones are unusual in biology. So pine trees know how to make pine cones, but this is still the only example in nature that we know at the molecular level of a cone. And so we wondered, how could that happen? And I'd love to tell you all of the details, but I'll just summarize what we were able to find. And we, as very talented graduate students like Barbie Ganser-Pornillis and Sam Lee, and also great collaborators like Chris Hill and John Finch. And what we were able to make conical capsids in the test tube, and we ended up deciding that their architecture must be that of an object called a fullerene cone. So this is an assembly that's named after Buckminster Fuller, the architect. He's the person who invented the geodesic dome, and it uses many of the principles that he elucidated in his architecture. And if we look at it, it sort of has two regions. It has a region in the middle that is a honeycomb. So you can see there are these 6-membered rings, and they're packed together just like a honeycomb if you open a beehive. But it also has to close at both ends. It's a closed cone. And in order to close, we have to have 5-membered rings, so-called pentagons. And there are 7 of those at the wide end of the cone and 5 at the narrow end of the cone. And this follows the same principle as an object we're all very familiar with, which is an old-fashioned soccer ball. So if you go and look at your black and white soccer ball, what you'll discover is there are 20 panels that are six-sided. Those are the white panels, but it's allowed to close into a sphere because there are 12 black panels that are five-sided. And the same principle is used by the virus to make the fullerene cone. So I need to then tell you that the next major advance was driven by Owen Cornelis, who's now a faculty member here at the University of Utah, together with Mark Yeager. And what they did was to take apart these fullerene cones and into their building blocks, and then study them at great detail using a technique called X-ray crystallography. And the details aren't so important, but what's important is that this was an advance that allowed us to see where the atoms in the capsid are. So what they discovered is the building blocks are in fact a hexamer and a pentamer. And that was important because it meant that the fullerene cone model must be right. The building blocks that were predicted from the model were was observed. But it was also important because we could now build atomic models of the capsid. We could know where all the atoms are. And that's important because drugs work at the atomic level. And so we need to understand atomic level structures in order to improve drugs. And that allowed Gilead to use that work to bind inhibitors to the hexamer. So lenacapavir, we'll see in a moment, binds to this site here in the hexamer. And they could look at how well an inhibitor bound and figure out how to make that inhibitor even better, even tighter. And that's the process that we call structure-based drug design. And it's critical because one typically starts with poor inhibitors, but hopefully through medicinal chemistry and structure-based drug design can end up with very potent inhibitors. And that's exactly what Gilead did. So I need to tell you what the capsid does. And so this is the first half of the virus life cycle. So this is now a cell, a T cell, a CD4 T cell being infected by HIV. So we're picking up where we left off in the animation that I showed you. We have an infectious particle, and now it's going to have to infect a new T cell. And there are lots of steps, and as basic scientists, we're interested in all of them. There's really only a few concepts that are important to realize. And one of them is that the virus has to replicate its nucleic acids. So it starts out with RNA, that's shown here in light blue, and through the process of reverse transcription, it makes a DNA copy of itself. And that copy is then integrated into the chromosome of the host cell. And this is the reason it's so hard to cure people of HIV, because every time a T cell gets infected, the virus makes a copy of itself and archives it in the infected cell. But the second surprising result was that we and others were able to show that throughout the first half of the viral life cycle, the capsid was playing very important roles. So the virus recognizes a CD4 positive T cell. That causes the envelope protein to fuse the virus and cell membrane, and that releases the core particle into the newly infected cell. And then, as I said, reverse transcription takes place, but we could show that it takes place inside the capsid. So it's important to have an intact capsid. And then others showed, to our surprise, groups like Hans-Georg Kreuzlich and Winnie Pothik, that the capsid actually goes all the way into the nucleus. And it's important for getting through the sort of door into the nucleus, which is called the nuclear pore. And so what we could show is even small changes in the capsid really mattered to the virus. And that suggested that perhaps the capsid would be a good drug target, because if you bound small molecules to the capsid, the virus should really care, because these different steps are all capsid dependent. And so that's exactly what has happened in the development of lenacapavir. So lenacapavir, one of its secrets is that it acts at multiple steps in the viral life cycle. So it can prevent the formation of capsids in the first place. It can affect different capsid-dependent steps. But the step at which it's most potent is as the capsid is getting into the nucleus. And this seems to be a vulnerability of the virus. It has to squeeze through this nuclear pore. As I said, other groups developed assays to show that it moves through intact. When lenacapavir binds, that makes the capsid very fragile. And so when it tries to do this, it fails, and that blocks virus infectivity. So in 2005, we were contacted by Tomasz Stilar, who's the vice president of virology at Gilead, and he asked if we thought capsid would be a good inhibitor, a good drug target. And of course, we were quite excited because a company like Gilead has the resources and the know-how to actually launch a drug development program. And so we helped them to start that, but I should emphasize that all of the drug development was done at Gilead. And this is a slide that I got from Tomasz sort of as a timeline. So I want to emphasize that this took Gilead some time, and it was a remarkable series of breakthroughs. So the things to remember are that the drug turns out to be wonderfully potent. So it's about 10 times as potent as any drug that we currently have. It's sort of frontline clinical use. And more importantly, it's long lasting. So a single subcutaneous injection of lenacapavir protects people from HIV infection for 6 months. And Gilead has just announced that an intramuscular injection can protect for a year. And so that's really very different from any tool that we've had available. And I just want to show you what Gilead had to do to develop the drug. So this is the drug that we know as lenacapavir, and it's quite a sophisticated piece of chemistry. As I said, they talked to us in 2005 and launched their program in 2006. And the first thing that they had to do was screen through lots of different molecules to look for molecules that might bind to the capsid. And that was directed by Steve Yant, an amazing effort. And they were successful after a couple of false starts, but that meant that they only had weak inhibitors. And so the next thing that they had to do in a medicinal chemistry program, which was quite amazing, directed by John Link, was to make more than 4,000 molecules and keep getting better and better and more potent molecules until they got to the molecule that they called then GS6207, which is the molecule that we know as lenacapavir. And of course, even at that stage, that just means that they had a good inhibitor. They had to, of course, show that it was safe and efficacious in humans. And they did that first in HIV positive individuals. And so it took 6 more years before it was approved for use in HIV positive individuals. It's turned out to be an important drug, especially for people who are resistant to the sort of frontline therapies. But along the way, they realized that its properties were really perfect for PrEP because it's so long lasting and so potent. And so this was the realization of people like Mopali Das and LGB that we're about to hear from. And so they launched this really remarkable clinical set of clinical trials that were very inclusive. You're going to hear about them that show that lenacapavir is really a very powerful PrEP agent. And I would just close by emphasizing how important that is. So worldwide, there are about 1.3 million new HIV infections. And so, of course, it's critical to treat the people who are HIV positive effectively, but it's also critical to prevent transmission. And ultimately, that could be a vaccine. But this really looks like an amazing new tool that allows us to prevent people who are in at-risk situations from becoming HIV positive. And with that, I'll stop and pass the baton.
Wes, thank you so much. Also, I'll just say your slides are beautiful. But a question before I turn to LGB. You showed the timeline from 2005 going forward with product development. I'm not sure I heard, when were the first activities that you did that
Yeah, so I
all the
maybe
things
said
we
that
saw?
briefly, but it probably went fast. We started our work in the early 90s. We started our lab in 1992. We planned it in 1991. So for perspective, that's the year Magic Johnson said he was HIV positive. The epidemic, there were not drugs, effective drugs. And so that's another, I can't do the math, but more than a decade before
Yeah.
the lenacapavir project.
Wow. No, that's really remarkable and amazing that you can condense 14 years of basic science into a handful of slides and then clinical development of 20 years into one. And with that, we will pass the baton. And although Wes is new to many of us on this call, LGB is not. Linda-Gail Bekker, although needs not use her full name, LGB, known to all, has been involved in pretty much every HIV prevention, and I should say most treatment and TB studies as well, that have happened, and has built not just an infrastructure to run clinical trials and to do implementation, but as you all know, has built a pipeline of investigators and collaborators in South Africa and around the world. And it is safe to say that there would be no PrEP if it weren't for LGB. I think she, and there are others in this community too, who've been involved in pretty much every PrEP, every microbicide, every vaccine study over the last couple of decades. And I don't know that you'll ever get zero infections again, as you showed in Purpose One. But you know, if for all those studies to have one that does that is a remarkable achievement. Of course, Linda-Gail was also past president of the International AIDS Society, amongst many, many other things. And I won't go on other than to turn it over to you, Linda-Gail, to walk us through what you were able to do with lenacapavir.
Thanks, Mitchell, and thank you to Wes. It's such a pleasure and honor to speak after him, and I guess to show just how this wonderful, science journey happens, all the way from brilliant scientists such as Wes making important discoveries and then handing off to us so that we can actually, do the delivery all the way into communities and people to change the trajectory of this epidemic. And it's been my singular honor to be involved in the lenacapavir journey. And I'm so glad to share this with others today who've also had that pleasure. But I'm going to share some slides. And then I'm going to turn off my video because we are also in Chile, Johannesburg here. I'm in the same office as Stacey at WITS RHI, where we're actually having a prevention trial design workshop. And because so I can turn off my video just in the interest of bandwidth and share properly and get on with this, because as usual, I have a few number of slides to get through, but I want to talk about my region and South Africa in particular as a pivotal country for the Purpose program. And just to point out that we are just one of the countries that have beyond the US, made huge contributions, I think, to both HIV, TB, and other infectious diseases, often at the generous and warm partnership that we have had with the US and colleagues and collaborators there, including the National Institutes of Health. And this has come under, as you said, under threat in the last couple of months, which certainly makes me a little tearful and dismayed. But I hope that some of this that I'm going to show you is just to show you the incredible legacy and infrastructure that has been built over a number of years. So these are some disclosures, just to say that I was national PI for Purpose One, and that we did have sites in both Purpose One and Purpose Two. Chris Byra, a very good friend and colleague, showed this extraordinary slide at CROI earlier this year, and it really just shows how hard, how intractable it has been to move incidence down, particularly amongst young women and adolescent girls in this region. And these are a number of studies. You see them listed underneath, either their placebo arm or actually the study themselves, where you can see the kind of incidence we've seen, not only way back, in the first part of this millennium, but all the way out to 20 years on. Just really an intractable problem for us to shift that incidence. And this led a few of us to write this rather doom and gloom question around, can we actually get to epidemic control by 2030? Not so much to say we're cynical, but to say what can we be doing and what is needed. And as Mitchell has said, I think we had some indication towards the end of last year when we realized that injectable PrEP might be a way to help us to get to that goal. And so I think we do stand at quite a crossroad about whether we are going to use these next 5 years to really begin to bend that arc towards zero. And this is the story of my region. We are a feminized epidemic in this part of the world. We contribute about half of those 1.3 million infections every year coming from women and girls in this region. And it amounts, if you want to think about this, to about 3,000 young women acquiring HIV every week in this region. So, Wes, if we go back to 1990, when you discovered the molecule, you will just be able to sum up how many young women and adolescent girls have become infected in this time. So although, you know, we recognize that science takes its time and it needs to come to fruition, every minute matters when it comes to preventing HIV. The other half of the infections happen in what we call key populations, as you all well know, and key populations occur all around the world. Globally, there has been some reduction in incidence. We've certainly seen it in sub-Saharan Africa, but half of the other 1.3 million infections are happening in people who sell sex, men who have sex with men, transgender people, people who use and inject drugs, and others who are incredibly burdened or vulnerable to HIV, but do not access services easily. And here you'll see up to 22% of people in my region are have a prevalence of HIV amongst transgender people, for example. So just drilling down to South Africa now, this was a pivotal study that Purpose One used to calculate where they needed to go for sites to ensure that we could run an efficient efficacy trial. And this study showed about a 4.5 incidence in a study that was designed to look at different kinds of contraception and a possible risk of contraception contributing to HIV risk. In fact, we showed this was not the case, but it was an important study for just showing what still needed to be done around HIV prevention. Here's another randomized controlled trial run in South Africa. It's called HVTN 702. It was a vaccine trial. It did not show efficacy, but here you can see the breakout of women and men. This was a study that enrolled both men and women, but women really bearing the brunt of the HIV epidemic. So we have been on this journey of ARV-based prevention all the way back to Vienna in 2010, when the Carims showed that tenofovir gel had some efficacy. But subsequently, South Africa, and here I thank Rafi for some of his wonderful graphics, but what I've circled is the studies that have participated in oral PrEP that have occurred again in this region. So sites set up as early as the early 2000s to not only look for better treatment, better TB therapies, but also to enhance the kinds of preventions we had available. And so we have participated in the dapivirine ring studies. I won't go into the details there. The cabotegravir long-acting 2-monthly injectable, and of course, Lenacapavir, this most recent 6-monthly, which has been so beautifully described by Wes. So this, recognizing the power of this molecule in the injectable formulation, it was undertaken that cisgender women and populations disproportionately affected by HIV incidence needed additional HIV prevention choices, and that we should design a study or a suite of studies that could test Len as a first-in-class multi-stage HIV capsid inhibitor with high potency and a long half-life as a twice-yearly subcutaneous injectable to see if it could reduce incidence in these populations. And of course, here are the countries and the places where both Purpose One and Purpose Two, the two registrational studies, were conducted. Purpose One for cisgender women, as you see, young women aged 16 to 25, 3 clinical trial sites in Uganda and 25 clinical trial sites in South Africa. South Africa did also participate in Purpose 2, and we did, offer some clinical trial sites, as you see in the diagram. Now, very importantly, this started well in advance of actually enrolling the first person. So partnerships were developed, and this is captured in a beautiful poster that was presented by Danielle Campbell at CROI earlier this year. Just the process of getting to this point. So there are partnerships with stakeholders, government, and importantly, community. Voices of community, the GCAG was formulated. We heard the voices of community advising how there should be a person-centric design, what was needed in the trial, what was the best way to go about it. And very loudly, there was the need for diversity, equity, and inclusion. And you can just see in the bottom panel how often advisor engagement occurred in a number of countries in a number of different ways to ensure that we did come up with a design that was in keeping with what people believed needed to be seen. And here you see the suite of studies. So the 2 registrational studies, Purpose 1 and Purpose 2, and then Purpose 3 being conducted as we speak amongst US cisgender women in the US, another study in the US amongst people who use and inject drugs. And then Purpose 5 is a Gilead-run study, which is being conducted in France and the UK in people also who could benefit from this kind of prevention. Just to say, Purpose 3 and Purpose 4 are both being run by HPTN. So again, the importance of the Division of AIDS in helping us to formulate these studies. Yeah, everyone is very aware now of how Purpose 1 was designed, how it looked. And this detail is available in the paper that was published at the time of Munich last year. Here's Purpose 2. It was published a little bit earlier this year. Again, a design that I think is very interesting, takes into consideration that we can no longer have placebo, true placebo. This is an active control study with background incidence in the endpoint. And now just looking at the baseline characteristics of both Purpose 1 and Purpose 2, you see a very wide array of individuals, all the way from age 16 in both studies to 25 in the Purpose 1, somewhat older in Purpose 2, but a population that really across the board that, judging by the numbers of STIs that are sexually active, and in parts of the world that could really benefit, as I say, from prevention. This is the historic sort of, for me, eye-popping results that we were for the first time ever able to achieve amongst young women and girls. We had seen very good results with cabotegravir with the 2-monthly, but really 0 infections in the active arm is unprecedented. Very close behind Purpose 2, just 2 infections in the active arm in that study. The reason, if you're asking why that was, well, we've always seen that young people, and particularly young women, struggle with daily adherence to oral PrEP. And this is borne out in the nested study, just to confirm that it isn't that oral PrEP doesn't work. It's just that these young people, for many, many logistical, personal, and other reasons, find it difficult to adhere to oral PrEP. But thankfully, 6-monthly injections seem to really turn that around. Based on wonderful literature starting to come through now about inclusion, particularly of pregnant and lactating people and younger people. The Purpose 1 study took this on board. The GCAG was very positive about needing to ensure that we were as inclusive of reproductive choice, of participation and support for these individuals. And so Purpose 1 included pregnant and lactating people, again, setting a new benchmark for how we do prevention studies. In addition, and by allowing people to be pregnant in the study if they so choose, although we did provide contraception for them, you can see there was almost 500 studies, pregnancies in this study, just underpinning how important this is for individuals. The inclusion of adolescents, again, a first for a phase 3 registrational study, and just showing that this is not only feasible, but it can be done, again, with partnership of community, of regulators, of ethical review boards, and the trial sites themselves. The involvement of adolescents has been a passion of ours, and in South Africa, there were a number of sites that, again, based on the fact that, and here's a study of ours just showing again that adolescents struggle to take oral PrEP, that we really wanted to see adolescents participate and somehow, if possible, be included in a label right from the get-go. And so we're very excited that the adolescent data will be going in, is with the regulators, and we hope that the inclusion of adolescents will be in the label should we see approval in the next little while. Important to do this kind of study because we were also able to show that not only was it safe and well tolerated, but the PK was the same in adolescents as adults. And we hope we'll also be showing PK data in breastfeeding and pregnant people later this year. Here again, just to confirm that plasma concentrations were quite similar amongst these populations. These studies also allow us to interrogate behavioral aspects. So here in our purpose study, we were able to ask them, well, what would you prefer, 2-monthly or a 6-monthly injection? Why would you choose between them if you had to? And we've heard all sorts of things from convenience. I'd rather stay with what I've familiarity, I stay with what I'm using. I'm in a 2-monthly injection program, or I'd rather go to 6-monthly because it might save me time. So starting to understand what users might prefer and what they would want for their lives, becoming part of the research that we're able to do. So 2 incredibly inclusive studies that I think do set a new benchmark. We were happy to participate in Purpose 2. You see some of our Pride events related to this. And you can see in our Purpose 2 enrollment, we really saw a nice array of everything from cisgender men to gender non-binary, transgender men and transgender women in Purpose 2, sort of in keeping with the SOGI universe that we uphold at Desmond Tutu. So these studies looking at a wide population of individuals, diverse types of people. And again, I think the Purpose program it has really set a new benchmark for how we do this in the future. Talking to access very quickly, what improves access? Well, there are a number of things that are important for access, but I would say that if you do the studies where you want to get the product in the future, it is very powerful because by providing solid evidence in those areas, by doing some of the implementation science in those areas, by having people experience them and speak to accessibility, safety, convenience, having the regulators look at dossiers in those areas, as well as, being able to impact cost and affordability by getting involved early on, and of course, demand generation. All of these things are improved when we participate in the studies. And just again, to make the point, South Africa has contributed to all of these studies over the many decades, including the implementation science. So here you see the CAB LA rollout and the importance of the region for doing the implementation studies. So very much, filling out where we are with PrEP today and hoping to keep contributing to the promise of new products coming down the pike. It reminded me, Mitchell, where we were in just 2019, just a handful of people in the region on oral PrEP. But over time, because we were able to be involved, to engage, you see this region making a huge contribution to the global PrEP uptake. And now we really are making the biggest contribution, or certainly had been doing up until some funding freezes earlier this year, but extraordinary how PrEP is now beginning to roll out. And I know you've spoken to the fact that regulatory is starting to happen now for LEN. Just point out that 2 countries that were involved in the registrational studies, as well as a number of others, are at the same time also looking at the dossiers, and we hope will soon follow the FDA, perhaps with the approvals. We are also looking forward to seeing the generics come through. And this is where, again, when studies are done in our region, we can start to have impacts on other important access advocacy, such as modeling, to understand what the investment case and how much this should cost, as well as the impact of rollout. And this is work by the HERO group in Johannesburg, who showed that if we can roll out LEN, we can actually eliminate HIV 10 years earlier than would have been the case had we not had LEN. So in summary, and I hope I haven't taken too long, I would just say that primary prevention that is taken to scale comprehensively, respectively, and equitably globally, wherever HIV is circulating, is the key to epidemic control. Effective primary prevention is most effective when it suits the user, and those users differ around the world. So it's important we go to where the users are. Longer acting, less frequently dosed is most effective in the young. I think Purpose 1 and Purpose 2 showed this. We will only see epidemic impact when prevention is scaled boldly. Scaling depends on access that is unrestricted and inclusive, and testing new prevention products where the burden is high is both efficient and appropriate and increases odds for accessibility. It also increases user, provider familiarity and enhances demand. And of course, these are more complex, they may be more costly to make, but it's really important that we now pay attention to getting them to everyone who needs them wherever they are in the world. And I'll end with that. I'll thank Wes for doing the basic science and for all the people who have funded the science to date. This has been incredibly important. We look forward to seeing the impact in the not too distant future.
LGB, thank you. I would not cut you off no matter the time. Your points are too important, and your summary is spot on. Thank you, and thank you both. And I know we don't have a ton of time. A lot of great questions have come in. I wonder if I could just start with a question to both of you. And again, as I said at the outset, there's a lot about Len and what we need to do to make sure we don't screw up what you all have delivered us in basic science and clinical research in terms of access. But I do want to ask about a different future, and that is the next wave of innovation. And if you could both just speak to briefly, what you're both experiencing, both Wes in your lab and in basic science with the assault on the NIH, to be perfectly blunt. And Linda-Gail, you mentioned the importance of the networks, not just for the purpose studies, but the PTN, the VTN, the ACTG, the clinical networks funded by DAIDS and NIAID, the PEPFAR cuts. What's it like for both of you as you think about the future and what we need to be keeping in mind, not to depress us, but to rally us?
I could start with the basic science end of the spectrum. That was a beautiful presentation, LGB. So one thing that I hope came out of our joint presentations is this concept that drugs go from bench to bedside to community, and that every step in that pipeline is critically important. And so speaking for the basic scientists, I think that basic science really has a remarkable history of having impact for human health. So I could give a million examples, but the obesity drugs were inspired by Gila monster venom, and the next revolution, which is going to be genetic engineering, came from learning how bacteria protect themselves against bacteriophage. And so what I think we view our role as trying to understand things so that we can think about them logically, and so that we can provide new and often unexpected therapeutic avenues. And so we can't promise exactly what we'll learn because we don't know it. But I think what history tells us is that basic science is a feedstock for new innovation. And so that makes it particularly challenging right now to think about the proposed NIH cuts. You may know that their proposal is, from the president's budget, is 40% cut in NIH and capping on something that's quite important called F&A. And I'm a department chair. This would really cripple our department, and it would cripple biomedical research across the US. And we have, I'll just finish by saying, you asked about timelines. One of the things that's really amazing is we can do in a day what took us a year. And so we're, just like Len is offering a new opportunity, so is basic research. We can just do so much more. And so these cuts are coming at a time when the opportunities, especially for our young scientists, are really amazing. So it's very important that we not do that.
Great. Thanks, Wes.
Yeah, and
LGBT?
I'll quickly sort of pick up on that and maybe talk about the investment. And the investment for me are in two tracks. The one is everything we have done with PEPFAR to date and where we are today. And then running right alongside, because I think the two are so incredibly interlinked, has been the investment in infrastructure and the clinical trial infrastructure that, you know, again, this is not a handout to us. We are very much in partnership with Wes and, you know, the NIH and collaborators in the North. And this incredible partnership has brought us to a point here where I would just say, Mitchell, you know, we've got 5 years to deliver on the promise of bringing this epidemic under control. And the light is at the end of the tunnel. And I don't say that lightly. I'm not talking about the end of AIDS. I'm talking about bringing the epidemic under control so that we don't see that incidence again. You know, we actually do begin to sort of bend that curve. And we now have in our hands the opportunity to deliver on that. And we've suddenly had this, you know, unbelievable sense of having the rug taken away from under us. But I would just say that, you know, my family, the people, ourselves, you know, we are not going to let this happen. We have to keep our eyes on the 2030 goal, and give ourselves, you know, maybe a little time to just regroup. And then we've got to get back on track. And that means innovation has to continue, and we have to deliver on the service, the treatment, the prevention, and, you know, bring these new innovations to bear. And that, I think, is just non-negotiable. I was stood at that point in 2000 where we were seeing graves and people dying every day, and we said, this is not okay. We're not going to let this happen. We're there again, and, you know, I think we just have to push through and make this happen again, that we do not see that we don't deliver on the dividends having had all this investment to date. And I would just encourage everybody on this webinar similarly to advocate, to get on board, roll up their sleeves, and just make sure we don't lose this moment, you know, because then this epidemic stretches out for time immemorial, which none of us want.
Yeah, no, we're not going back. Linda, I wonder, you know, you were hosting both Purpose 1 and Purpose 2, the only country actually that hosted both Purpose studies. There's a lot of talk right now coming from the NIH. We don't yet know officially. We're told a new policy is coming in September about international research. Can you, well, I'd love for both of you, can you imagine what would have been the time frame to answer the question, did Len work, if we didn't have sites in South Africa? Could we have answered the question, or how much longer would it have taken?
So 25 of the 28 sites come out of South Africa. You know, the bulk of the incidence comes out of South Africa, and that's for HPTN 083, 084, Lenacapavir. It's our, you know, wonderful aspect that we have these experienced FDA accredited clinical trial sites, but we also carry, it's one of the biggest burdens in the world. So, you know, plus we have the labs and the safety and the infrastructure to support. So it's a beautiful marriage in heaven in a way that we really can bring this together and bring out highly efficient studies. And this is the same for TB. It's the same, you know, for a number of sort of conditions. And that also is just, you know, so important to recognize what can be lost, because this has been built over 30 years that I've certainly been, you know, at it. And it can be, Mitchell, in the next, unless a miracle happens, of the 400 highly experienced staff that I have in my 7 sites in South Africa, I will have to retrench 1/3 of them. 130 people. These are, you know, highly trained clinical scientists who are going to have to look for other jobs. And I say this not because I'm looking for sympathy or anything. I'm just, this is experienced expertise that we are losing, you know, and that makes me very anxious indeed, because in the same way that we were able to pivot very quickly during COVID-19, there's still work to be done in HIV. There's certainly still work to be done in TB, and there's much work to be done generally, that we have those products that Wes is talking about. We are an ideal place, along with other countries, but certainly my country that, is out in the cold at the moment, we have a contribution to make.
Yeah. Incredibly helpful. Linda-Gail, there's a question in the chat that I think is important, because we're used to so many of these PrEP studies and vaccine studies frankly, since the beginning of prevention R&D, that were either funded entirely by the NIH or co-funded with CDC, USAID, the Gates Foundation. So someone asked the question, who financed the PURPOSE trials, the PURPOSE 1 and PURPOSE 2? And I think it's important for maybe you to clarify that. But also, what could that have happened without the infrastructure? The networks didn't do it, but the infrastructure existed because they did. And I wonder if you could just really quickly speak to that.
Yeah, I think that's important to say. So that the Gilead program was run by Gilead, and with lots of help from friends and family, in terms of, we do this as AVAC was involved, other groups were involved, coming along, making sure the forum, etc. I mean, there are a great number of people who made contributions. The actual per participant costs were covered by Gilead, but what I've tried to illustrate here, these are the sites that get used time and time again. They were put in place, many of them, by the Division of AIDS or the networks, and they are there, multi-purpose. They're available. They're multi, expert type responses. Some of them are doing TB as well as vaccines. Some of them are doing non-communicable diseases. But that infrastructure was certainly put to good use by Gilead. And we're at people's disposal for that, in that regard, in order to move the science forward. So quite right, it was a Gilead sponsored study.
Great. No, thank you for that. I wonder, but to both of you, but Wes, there's obviously a lot of excitement about LEN, and I think for all good reason. It was a little alarming a couple of weeks ago when the NIH basically cut the major HIV vaccine discovery program, the CHOV-Ds, one at Scripps Research and one at Duke. And the official announcement from the Department of Health and Human Services, repeated by Jay Bhattacharya, the new head of the NIH, in yesterday's Senate hearing, he specifically said, we don't need to do that research anymore because we can end the epidemic with LEN. Love the focus on LEN, but I wonder, as a basic scientist, when you hear Jay Bhattacharya, the head of the NIH, say that, do you get excited because you helped create the pathway to LEN, or does it run shivers down your spine?
I guess I would say the latter. So I want to be a little bit careful, because I think we should be positive going forward. I don't want to fight. I want to have things succeed. But I was on the advisory board for the Duke CHOV program. These are amazing scientists, and a vaccine is a different beast than a long-acting PrEP. And so the idea that we don't have a use for a vaccine is just not correct. And I think that there are these beautiful papers in Science showing that it's possible to direct the human immune system toward neutralizing antibodies. This is a really hard problem, but there's now real progress being made. And so it is hard for me to see people I value a lot, but also people who are making major contributions lose their funding.
Yeah, no, thank you.
And Mitchell, I would just say, yeah, I would just add to that. I was very careful to say that we can bring the epidemic under control as a public health threat by 2030. We will not eliminate this. We will continuously have to be watching our backs, even with as wonderful a molecule as lenacapavir, because it requires people to come back. It's a pharmaceutical that requires people to come back every 28 weeks, and if they don't, it will not work. And that is the downside, if you like, of these pharmaceutical agents, and it's the beauty of a vaccine, should we be able to find a vaccine that is effective and safe and affordable and accessible in the future.
Yeah, no, I really appreciate that, Linda-Gail, and I was really pleased the way you framed that. And I think language matters a lot. I mean, more than ever. And actually, I wonder, and I was just about, I don't think I'm calling her out now because she just posted in the chat. I was going to see if Wes, your co-recipient along with Mopley last week of the award, Yvette Raphael's on. Yvette, I wonder if you might want to just jump in as, like me, a non-scientist. Um, and I thank you for, they
While
are amazing
she's jumping
presentations.
in, I'll just say what an amazing contributor she and other community advocates are. I mean, the reason these trials are completely different is because of that community.
Yvette, did you want to jump in?
Hi, Mitchell. Hi, everyone. Thank you so much. Great presentation. And to just see the connection between LGB's presentation and West's humbleness. I posted in another group to say you'll never see Wes as a genius, because that is how I describe him, because he's so humble. Great to
My
listen
kids
to
can
you
tell you
again.
differently.
Even my kids don't think I am that genius. But I just want to say, unless we do something real quick, the gains that we've made will be irreversible. And I understand LGB when she talks about just how painful it is to lose the brain power. The reality on the ground is for us to rebuild will take as much time as it was to build for us to where we are today. And we do not have that time. HIV will not be the inheritance of our children. I refuse to believe that. So we need to fight, and we need to fight hard. And if it means impeaching some people, it's okay. Have a great afternoon, everyone.
Yvette, thank you so much. I know we're just a few minutes over. I do want to, and we have to close up. And I've come to our Granch's question in the chat at the end, but I want to give both LGB and Wes a chance to last words, and perhaps maybe what's your best hope for the next, say, month of headlines? Len's going to get approved. What would success look like over the next month or two to both of you? LGB, do you want to start?
I'd love to know that NOAs are open again and flowing. That would be really notice of award, so we can keep doing the work we were doing. That would be just brilliant. But also to say that, we know that there are 10 million doses of Gilead, of Lenacapavir injectable PrEP that is going to be available. I'd love to know that is purchased and is going to the people who need it most, and that we're going to save some infections because of that. That'll be just wonderful.
Love it. Wes?
You don't have much to add, but in the basic science community, it's what LGB said is true. We have 5 grants in our department that have been canceled and others that are being delayed. And these are people who are tackling problems in metabolism and problems in cancer. And so it's as important as HIV is, it's across the board in human health. And I would also say that, the same investment story is true for us. These people have been invested in, they're enthusiastic and incredibly talented. And so they need the resources to finish their jobs.
Great, Wes, thank you. I want, to our Granch in the chat, I want to be clear, as far as we know, Gilead is not cutting funding. The US government is, but but others are too. We are now moving into a moment of the Global Fund to be replenished. Global Fund is meant to step up even more into the Lenacapavir rollout space. They're going to need to be replenished. And we will do additional calls as AVAC about what is the advocacy, but maybe I'll just say really quickly, Len matters. And what Linda-Gail just said, there'll be millions of doses produced by Gilead. The hope was that PEPFAR and Global Fund would both procure. We don't know what that's going to look like in the future. So advocates need to engage and not just celebrate science, although it's celebratory, not just celebrate a regulatory approval, but we need to push every donor and every government to recognize this is an opportunity not to be missed. So Len matters. The other thing I'd say is basic science matters, because we are far from done. Huge progress, but if the next Wes doesn't have an idea 20 years from now, we're not going to have a product going into clinical development, and 30 years from now, we're not going to be celebrating the next big innovation in any therapeutic area. So basic science matters. And then third, Linda-Gail, thank you, because this, we all know this on this call, but I think we need to be louder and prouder advocates that all of this depends on global partnerships. We say it all the time, but this gives us, I think what you described today, this gives us a meaning that with that, women in America will get lenacapavir because of women in South Africa and Uganda. That's how America gets safer, stronger, and more prosperous. And that research couldn't have been done in the US alone, which I know you all know, but I think we need a clarion call that Len matters in the short term, basic science matters, those global collaborations matter. And I really just want to thank both of you for helping tell that story, because it's not about a single product and it's not about a single disease. It's just such an amazing case study for what science can do. I must say, I have never been more excited about science than the last 65 minutes. So thank you both for doing that. There are many, many calls to action for all of us. Stay tuned for more, lots at avac.org. I just want to thank everybody for making time and most of all for committing to be great advocates, whether you're a PhD, an MD, or just matriculated. All of us need to be science advocates going forward. Thank you. I want to thank Stacy Hanna at AVAC and Levi Butcher at AVAC for helping pull all of this off. It takes a village. And thank you all for joining and more to do, but thank you both.
Thank you, Mitchell.
Cheers, everybody.
Bye.